do well to indicate in the comment box below. Say I can hear you. All right, this is going to be an audio class, and of course, we are going to be focusing on MCB exam areas. All right, areas that are actually more commonly focused on when it comes to MCB 200. Okay, like I said, if you can hear me, please type "I can hear you, sir" in the comment box so we can proceed immediately. All right, I can see your responses. I can see your responses. Good, good, good. Okay, so basically, we are going to be kicking off from where we stopped in the last class. All right, so just like we did last time, we'll be focusing on major topics. All right, major topics. I'm going to be explaining the topics. Then I'm also going to state some questions that are commonly asked. All right, some commonly asked questions in those areas that we are going to be treating. Okay, so um, yesterday we were able to go to a large extent by doing justice to the introduction to microbiology down to the aspect of the industrial microbiology. All right, so you're welcome. I can see you, Divine. I can see you, ebook vibes. I can see you, Bright Faith, Echo by God's power. I can see you, Richard, Tamuno Ibiomi. I can see you. I can see you all. All right, so you're welcome. You're welcome once again. So I want you to stick to the end of this class. All right, stick to the end of this class, and please pay attention. Get your pen, get your writing materials, and take down notes. Okay, take down notes. All right. So basically, like I said previously, you have to take note of the history of microbiology. All right, the history of microbiology, and of course you have to note that microbiology is defined as the study of microorganisms. Okay, and many scientists has actually brought about different inventions. Okay, we talked about Antonio van Leeuwenhoek, who happens to be the first to discover microorganisms and observe living microorganisms, and he called them animalcules. We said that. We talked about Robert Hooke, who discovered cells in 1665. We talked about the first to describe microscopic structures, Louis Pasteur. Okay, we talked about the father of modern microbiology, who happens to be Robert Koch. All right, although the father of microbiology usually credited to Antoine van Leeuwenhoek. Okay, so Robert Koch also talked about the Koch postulates. Okay, Louis Pasteur um, has a certain level of credit when it comes to the development of modern microbiology. He has a high level of credit when it comes to the discoveries of microbiology in modern times. Okay, So we also have the the first person who developed vaccine. That is the first vaccine to be developed. That was for, I think, smallpox. Okay, that was Edward Jenner. Edward Jenner was the one who discovered or developed the first vaccine, which was the vaccine that could cure smallpox. We also have Paul Ehrlich. Paul Ehrlich, who happens to be the father of chemotherapy. We have Alexander Fleming, who discovered penicillin. All right. Okay. So basically, when we talk about microbiology, we cannot talk about microbiology without talking about abiogenesis and biogenesis. All right. So basically, they are very important concepts, but biogenesis has to do more with microbiology because it's talking about life from existing living organisms. All right, life from existing living organisms, and how microorganisms actually develop and originate from existing organisms. Okay, from existing organisms. So it's very crucial, it's very important to take note of that. All right, it's very important to take note of that. Now, one thing we have to also understand is that most of the questions that you would see in micro introduction to microbiology examination is basically based on topics that you are familiar with. That's why we are trying to ensure that we familiarize ourselves with these topics. Okay, with these topics. All right, if you're joining, okay, I can see someone joining today. You're welcome. You're welcome. It's all right. So now let's proceed. Let's look at some very repeated questions, some very repeated questions on this introduction. Okay, this introduction. Okay, so there's a question that I saw one time. It states, until the 17th century, advances in microbiology were limited to dash. Until the 17th century, advances in microbiology were limited to dash. All right, so what actually stood as a limitation, okay, for advancement in microbiology until the 17th century? That is how come, it was the 17th century now that microscopy and microscopes were discovered, cells and all of that. So what actually caused the limitations? What was happening all those while, 16th, 15th century and all of that? So what happened? It was actually due to the lack of appropriate tools to observe microbes. All right, the tools that were available were not appropriate to actually observe microbes until the discovery of the microscope. All right, so microorganisms were not even so prevalent, we are not prevalently known. All right, we were not prevalently known until the invention of the microscope and until Robert Hooke identified cells under the microscope. Okay. All right. So another question that is also commonly asked: in 1665, Dash discovered small boxes called cells. Small boxes called cells. The answer is Robert Hooke. Robert Hooke, what he did was fantastic, very phenomenal. So he took a cork. He took a cork, like, cork now, like that wine top that you remove and it makes a particular sound. Okay, so he took something like that and he sliced it into pieces, and he took one piece and placed on the slide under the microscope, and he saw small boxes like compartments, and he called them cells. All right, that was actually done in the year 1665. Okay, then another question that is commonly asked: Anthony van Leeuwenhoek is called the father of microbiology because of dash. Because of dash. So why is he called the father of microbiology? Because of dash. Now the answer is that because he discovered living microorganisms in pond water. All right, he discovered living organisms in pond water. Pond now. So that was where he actually discovered living microorganisms. Okay, living microorganisms. Now then we also have another question: abiogenesis states that dash. Now, what does abiogenesis mean? Abiogenesis states that microbes arise from non-living things. Microbes arise from non-living things. Okay, but that is not entirely correct, because microbes arise from living matter. Okay, microorganisms arise from already existing living organisms. All right, that is biogenesis. That's biogenesis. All right, then another question is, medical microbiology covers dash. Okay, now these questions that I am pointing out to are repeated questions or frequently asked questions under the introductory aspect of the general microbiology. All right, so medical microbiology covers dash. All right, so it covers pathogens, immunity, and epidemiology. Pathogens are actually disease causative agents. Pathogens are things that causes diseases. Immunity is fight or response to diseases. Okay, being immune is having a particular response to diseases and illnesses. Then epidemiology is actually the study, when you're studying diseases, studying the behavior of diseases, studying the spread of diseases. Okay, studying the spread of diseases and all of that. So that's what medical microbiology covers. All right, so then another question that you see commonly is, who established fermentation? In yesterday's class, I explained fermentation, and I talked about fermentation as an importance or significance of microbes. Microbes are used to ferment, like yeast. I told you that fungi, which is a microbe, fungi like yeast, is actually used for fermentation. All right, microbes are also used for, I told you, for preservation of food substances and all of that. All right, so who established fermentation? It was Louis Pasteur. Louis Pasteur. Okay, Louis Pasteur. Pasteur is spelled Pasteur, not the pastor in church. All right, Louis, Louis. All right, Louis Pasteur. He was actually credited to have established fermentation. All right, then another question that is commonly asked is, state the germ theory, or the germ theory states that dash. Germ, like germs, like GERM. Germ theory states that dash. Okay, the germ theory states that diseases are caused by microorganisms. Very simple. That's more like the simplest or shortest theory in microbiology. All right, it states that diseases are caused by microorganisms. That's the germ theory. All right, so I stated earlier, I told you that the father of immunology is Edward Jenner, and I also told you that the Nobel Prize for tuberculosis research, which was in 1905, it was awarded to Robert Koch. All right, he was actually a scientist who, um in modern microbiology, that's why sometimes it's usually between Pasteur, okay, Louis Pasteur, and Robert Koch. Sometimes there's usually an argument who the father of modern microbiology is and all of that. All right, because Robert Koch did fantastic stuff, so phenomenal stuff, wonderful and amazing things. All right, he was the one who did the largest research for tuberculosis, largest research that even got to Nobel Prize in 1905. All right, so that is that. Okay, so don't play with the names of these scientists. All right, Leeuwenhoek, I told you microbes. Robert Hooke discovered cells. Then Koch discovered diseases, Pasteur fermentation, Ehrlich chemotherapy. Okay, father of chemotherapy. Fleming was the one who discovered penicillin. All right, so don't forget. Okay, so don't forget too that biogenesis is the true fact against abiogenesis. Life comes from life does not come from non-living things. All right, a wood cannot produce a living thing. All living things comes from living things, and that's biogenesis. All right, and that's biogenesis. All right, so that's a very huge fact that you must take note of. All right, so basically, I mentioned the different types of microscopes last night, okay, in the last class, but I did not give you the significant differences. Okay, so of course, remember that microscopes are used to observe microorganisms. Without the invention of microscopes, probably there wouldn't have been any advancement in microbiology. There wouldn't have been any advancement in microbiology. All right, so some key terms that you must take note of is magnification. When it comes to observing microorganisms. If you don't properly magnify them, you may not be able to see the cells or be able to see pathogens that are present in a particular sample. All right, so magnification is important. What is magnification? The process of increasing the size of a particular image, the size of an image. Okay, so that's what magnification is. All right, so we also have resolution. Resolution is ensuring the clarity of image, making the image that you're viewing to be clearer. Okay, so when you are ensuring the clarity, okay, by increasing the resolution, resolution is making that image to be clear. Okay, increasing the magnification is making the size of the image to be larger. All right, so I believe you can differentiate both of them. Okay, so I talked about the types of microscopes. I said we have simple microscope and simple microscope. The one thing about simple microscope that you actually see and you will know that this is a simple microscope is that it has one lens. It has one lens. Okay, now the compound microscope has multiple lenses. Okay, multiple lenses, that is two or more. That's a compound microscope, two or more lenses. Then an electron microscope can have two or more lenses. Okay, but the reason why you cannot call an electron microscope a compound microscope is that the resolution of an electron microscope is higher than that of a compound microscope. That is one thing that is unique about electron microscope. That's why if you even go to some organizations, in fact even in the University of Port Harcourt, there is only one electron microscope. There is only one electron microscope. I don't even know if it's still there or they've moved it out. But electron microscopes are very, very expensive. They are very expensive because of their high resolution. All right, their resolution power is very, very high. Okay, very, very high. So that's it. Okay, so that's it. So take note of that. Take note of these types of microscopes and all of that. All right, so now we would also look at bacteria. Yesterday we looked at the structure and morphology of bacteria. All right, so but I didn't give you definition, and I said okay, I'll give you definition in this class. And what do we define bacteria to be? So bacteria are prokaryotic organisms, which means that they lack a true nucleus, and they also lack what we call membrane bound organelles. Okay, when I say true nucleus, I'm talking about the defined nucleus. Remember I said that cells are divided into two. We have the prokaryotic cells and we have the eukaryotic cells. The prokaryotic cells are those ones that do not have a defined nucleus. Defined nucleus is like a visible nucleus. You cannot visualize the nucleus of a prokaryotic cell. It's almost not there. It's almost not there. But the eukaryotic cell has a very clear and visible and defined nucleus. All right, so bacteria are prokaryotic microorganisms. All right, and their structure is simpler than eukaryotic cells. All right, but one thing about what's it called, the bacteria is that when compared to eukaryotic cells like that of plants and some kind of fungi, is that bacteria has very high efficiency for survival. All right, bacteria can survive in different conditions. I said something about that yesterday. We have cold conditions. Okay, we have some hot conditions and all of that. So basically the survival rate for bacteria the survival rate for bacteria is higher. So that they are prokaryotic cells that you would say it doesn't have a defined nucleus, it's in a way their structure is actually helping them. Their structure is highly helpful, highly beneficial to them. All right, so it's very important to note that their rate of survival and even how they reproduce. That's why bacteria spreads very, very fast, because their level of reproduction is very, very efficient. Just imagine that is humans that reproduce the way bacteria reproduce. uh you see lots of children. In fact, the world population will be quadruple, will be times four of how it is now. All right, if humans were reproducing the way bacteria reproduce, the the reproduction rate of bacteria is very, very rapid. All right, so I believe you can you still hear me? Let me ask now, so it doesn't seem as though I'm talking to myself. If you can hear me, say I can hear you, sir. If you're still with me, say yo, I'm with you, sir. I want to see your responses. If you can hear me, say I can hear you, sir. Okay, okay, okay. Okay, one response, one response. If you can hear me, say I can hear you. Okay, all right. All right, all right. Good, good. Let's proceed. Let's proceed. Let's proceed. All right. Now, moving forward, we have the basic structure. You need to understand the structure of bacteria. It's very important, it's very crucial to understand how bacteria is structured. There's what we call the cell envelope. All right, the cell envelope. Now, this is actually the outer covering of the bacterial cells. All right, the outer covering of the bacterial cell. Just like normal cell, we have the cell wall or the cell membrane in the animal cell. That's how the outer, this one is like an outer covering. Okay, it's not really the cell wall because we still have cell wall too, but this one is like the outermost covering of the bacterial cell. It's called the cell envelope. Okay, so the cell envelope has the cell envelope that now has the cell membrane and the cell wall. Okay, it's the cell envelope that has the cell membrane and the cell wall. Okay, so it includes two parts, the cell membrane and the cell. So the cell membrane is also called the plasma membrane. All right, the function is to ensure that there is what we call selective permeability. Things do not just enter the bacterial cell like that and come out. That's why the cell membrane is also called a semi-permeable membrane. A semi-permeable membrane. All right, so and also this cell membrane helps in the transport of nutrients being transported, proteins, carbohydrates, lipids, and all of that. Water too. All right, it's being transported via the cell membrane. Okay, and you know that there are also some cells that needs active transport. All right, so the active transport would imply that they will need what we call energy production. Yes, energy production. I talked about that yesterday. Energy production by virtue of production of adenosine triphosphate, which is ATP. All right, so because of that, they actually produce energy. They can actually produce energy, so there can be active transport. So understand that. And then for the cell wall, which is still part of the cell envelope, the cell wall is what maintains the shape of the cell. Okay, what maintains the shape of the cell, and it provides protection. Okay, it's a rigid layer, very fixed layer. I said it yesterday. It's a very fixed layer, and it's made up of what we call peptidoglycan. That's the component of the cell wall. Peptidoglycan. And there is also a part of the cell wall that helps the cell wall to actually remains the way it is rigid. All right, and that's a complex carbohydrate called cellulose. Okay, remember I said it has peptidoglycan, it also has what? Cellulose. Okay, so the major function of this cell wall guy is to ensure that the bacterial cell is protected. Is to ensure that the bacterial cell actually maintains its shape. The cell membrane has no business with the shape of the bacterial cell. The cell membrane ensures that there is transport of nutrients are being transported. It ensures that energy is being produced. It ensures that there is a selective permeability. All right, not anyhow liquid or anyhow substance can actually enter and leave the bacterial cell. Okay, so that's one thing you need to know. All right, so when it comes to this cell wall, all right, when it comes to this cell wall, the kind of bacteria actually affects the type of cell wall. I want you to understand that. There's what we call gram positive bacteria. Remember I talked about gram now. Yeah, it's even one thing. Say that Hans Gram, I mentioned Hans Gram that invented gram positive and gram negative bacteria. He was the one that discovered. He named it after himself. All right, so basically, like I said something earlier and I'm going to repeat it. When it comes to the cell wall actually varies based on the kind of bacteria. Okay, we have the gram positive bacteria and we also have the gram negative bacteria. Now for gram positive bacteria, let me tell you one thing that is peculiar about the cell wall. That peptidoglycan I talked about is very thick in gram positive bacteria. Okay, in gram positive bacteria, and there is a stain. The stain that is actually in a gram positive bacteria is purple in color. That is the stain. The stain is, you know what we know now. We'll talk about staining, shall we explain staining in details. But just understand that the kind of stain that is in gram positive bacteria is purple in color. But for gram negative bacteria, the peptidoglycan is not thick, rather is thin, like THIN. It's thin peptidoglycan. Okay, it's thin peptidoglycan, and the stain there is pink or red. Sometimes it can be pink, sometimes it can move from pink to being red. So it's either pink or red, but the stain for the gram positive bacteria is fixed, it's always purple. So don't forget that, don't ever forget that. Okay, so we are still talking about the structure of bacterial cell. I've talked about the cell envelope, right? I've talked about the fact that we have the cell wall and all of that, but we have what we call external structures so, as well. All right, now we have the flagella. All right, or some people pronounce it as flagella. Okay, we have the flagella. I talked about it yesterday. I said that it's used for movement. All right, bacteria uses flagella for movement. Okay, and another thing is that many bacteria move with flagella like as though it's a tail. Okay, they move with it like a tail, the way lizard has tail and crocodile has tail and all of that. So they move with it like tail. Just understand that flagella is used for movement. It aids motility. Okay, it aids motility. Okay, it aids motility. Then we also have another structure which we call pili, pili, which is also known as fimbriae. Fimbriae is spelled Fimbriae. Okay, pili, fimbriae. Okay, so what is actually the function of this guy? All right, so it's actually used for attachment. Okay, it's actually used for attachment, not the attachment on hair. Talking about the attachment of bacteria. So bacteria attaching to itself or attaching to substances. Okay, so the fimbriae enables bacteria to actually attach to other substances, to attach to surfaces. Okay, it's just like a glue. It makes it to fix itself, it makes it to attach itself to surfaces, makes it to attach itself to other organisms as well. All right, remember I told you that bacteria and most microbes tends to diffuse into surfaces. All right, so I want you to bear that in mind. Okay, so they are called fimbriae. Okay, and they are short too. They are not like flagella that is as like long, looks like tail. All right, these ones, they are short, they look like hair. They are hair-like structures. Okay, they're hair-like structures. You can still go online and check the structure of the pillar. You see that they look like hair. They are hair-like structures and they are short. Okay, they are short, just like a short wig. Okay, that's abstract though. Now let's move forward. Okay, then we also have what we call the capsule. The capsule is also an external structure. All right, it's called glycocalyx. Glycocalyx. Okay, glycocalyx. Glycocalyx. Okay, that's the capsule. All right, so it's like a gel, like gel-like. It's gel-like in texture. All right, so what it does is that it protects the bacterial cell as well. All right, it protects the bacterial cell. It prevents the bacterial cell from losing its nutrients, and of course its substances, internal substances that facilitates the survival of the bacterial cell. Okay, the survival of the bacterial cell. So that's all for the basic external structures. Then how about internal structures? We have the cytoplasm. All right, the cytoplasm is it's also jelly like, but it's not as thick as the capsule. All right, it's not as thick as the capsule, but it's also jelly like. All right, so it contains enzymes and nutrients. That's where all the proteins, amino acids, of course, the vitamins, the lipids. That's where they are encapsulated. All right, so it contains nutrients. It contains enzymes as well. Enzymes that speeds up the rate of the chemical reactions and all of that. All right, so we also have internal structures like ribosomes. Okay, ribosomes as well. So ribosomes are actually those organelles where protein is synthesized, where protein is produced, where protein is formed, and all of that. Okay, so but the ribosomes in bacterial cells is actually smaller than that of your own ribosome. Okay, it's smaller than your own ribosome, and your ribosome, my ribosome, is actually in eukaryotic cells. Okay, because we are multicellular organisms. So all multicellular organisms have eukaryotic cells. Most unicellular organisms have prokaryotic cells. Okay, so ribosomes is actually important because when the cell starts degrading, when the cell starts diminishing in its property, the ribosomes tends to produce proteins that will help to replace the worn out part of the cells. So you know that proteins, what proteins does is that proteins replace damaged cells and tissues and all of that. All right, so we also have what we call the nucleoid. Now, the nucleoid contains bacterial DNA. Remember that bacteria is a prokaryotic cell, so it does not have a nucleus. So now you may be asking, where is DNA? Because normally DNA is usually in nucleus. So DNA is a genetic material, right? DNA and RNA, they are genetic material. All right, they produce genetic information. So if you are hearing now that bacterial cells are prokaryotic cells and they do not have a defined nucleus, where then is DNA? DNA is actually in a nucleoid. Nucleoid is spelled Nucleoid. Nucleoid. All right, so it's actually where the DNA of the bacteria is. It's in a nucleoid. All right, so now moving forward, we also have the what we call the plasmids. Plasmid is spelled Plasmids. Plasmids. Now, what are these plasmids? They are actually carriers of genes. They're actually carriers of genes, and they do what we call antibiotic resistance. All right, so these plasmids are responsible for helping the bacterial cell to resist antibiotics. Okay, to resist antibiotics. So it is these plasmids that helps the bacterial cell to resist the amoxicillin you drink when you want to cure a particular bacteria, or maybe any other antibiotics like ampiclox. All right, so these plasmids are there to resist it, because bacteria do not like antibiotics. Now, it's antibiotics that are like the major treatment against bacteria in the body system. All right, so plasmids are there to resist antibiotics. All right, so that is that. All right, so I talked about diplococci yesterday when I talked about bacterial arrangements. Okay, so there's what we call diplococci. Okay, there's what we call streptococci. We also have staphylococci. Okay, some people pronounce that cocci as cocky from staphylococcus aureus. All right, so staphylococci, streptococci, and diplococci. Now, diplococci is actually when bacteria are in pairs. When bacteria is actually arranged in pairs, that's what we call diplococci. Diplococci is spelled cocci. Okay, cocci. Okay, then we also have what we call streptococci. Streptococci is when the bacteria is arranged in chains of bacteria. Then we also have like the common one, which is staphylococci. Staphylococci is when it's in clusters, when it's clustered together. That's staphylococci. That one is the most dangerous, so don't play. That one is the most dangerous. All right, it's more dangerous than diplococci and streptococci. Okay, so this one that is in clusters is very dangerous. Okay, so that's the point. So there are some questions that are commonly asked when it comes to bacteria. Okay, I would tell you some of these questions so you can take note of them. One question is, which of the following describe curve curved rod shaped bacteria? That is, which of the following options have a curved rod shaped, that is a rod shaped but is curved, a curved rod shaped bacteria? A, is it cocci or bacilli or spirilla or vibrio? Is it cocci, bacilli, spirilla or vibrio? Now the answer is vibrio. All right, because vibrio is comma. You know how comma is now, comma shaped. That's how vibrio is. You can do your research, you will see that the shape of vibrio bacteria is like comma. Okay, so that comma is called curved rods, like rod now, but it is curved. All right, so they are curved rods. All right, so the answer to that question is vibrio. So it's vibrio that describes bacteria that is a curved rod shape. All right, then another question is, staphylococcus bacteria are arranged in dash. Staphylococcus bacteria are arranged in dash: A, chains; B, clusters; C, single cells; D, spirals. Remember, I talked about staphylococci. Okay, I talked about streptococci, I talked about diplococci. I talked about diplococci, that they are usually arranged in pairs, streptococci in chains, and staphylococci in clusters. So now the question is about staphylococcus bacteria. They are arranged in dash: A, chains; B, clusters; C, single cells; D, spirals. Okay, so the answer is clusters. All right, so if you see the staphylococcus bacteria shape, it looks like, how will I put it, grape, like grape like. Okay, you know how grape is now. You know how grape is not really like orange. Grape is not really like, it doesn't have really a smooth, surface like that of orange. So it looks like grape like structure, clusters. That is the structure of staphylococcus aureus, look like grape like clusters. All right, so another question that is very very important is, which bacteria are coiled and spiral shaped? Which bacteria are coiled and spiral shaped? Now you see this question, this question comes in many years. It appears in a lot of years. You have been going through your past questions, you see this particular question a lot. So which bacteria are coiled and spiral shaped? Okay, I told you initially that the ones that are spiral and coiled, they are called spirochetes. Okay, I mentioned that in the last class. Spiro is spelled spiro. Sheets is spelled chetes. Spirochetes together. All right, they spelled together. Okay, spelled together. All right, so we have spirochetes. Okay, then now moving forward with another question. The outermost structure in bacteria is dash. A cellulose; B, peptidoglycan; C, cell envelope; D, mitochondrion. The answer is cell envelope. Remember, I told you that the cell envelope is the outermost structure in bacteria that has the cell wall and the cell membrane. Okay, cell wall, cell membrane. You also have the capsule. You remember what I said about the capsule, right? What did I say about the capsule? If you remember, I told you that it protects the cell. Okay, it's also gel like. I said it's gel like, but when I talked about cytoplasm, I said cytoplasm is internal, but cytoplasm is not as thick as that of the capsule. The capsule, I told you, don't forget to, the capsule, I said, is also called glycocalyx. All right, glycocalyx. Okay, so one more time, if you can hear me, say I can hear you. If you can hear me, say I can hear you. Before we proceed, if I can hear me, I have to be doing all these checks once in a while, so I would be sure that I'm not talking to myself. All right, okay, someone is still active. I can hear you. Okay, okay, who is that? Bright. Okay, Bright. Divine, I can see you. Okay. All right, good. Good. Okay, now moving forward, we also talk about bacterial growth and metabolism. All right, although I emphasized on it in the last class, but I will be more emphatical in this time. I will be more emphatical and more detailed about bacterial growth. Okay, so basically, bacterial growth is not just growth in size. Bacterial growth does not have to do. It's not the size of growth in the size. Okay, bacterial growth is the increase in the number of cells in bacteria. That is increase in number of bacterial cells. It does not have anything to do with the increase in the size. Please just note that. So in case they ask you if bacterial growth involves the size, you will say false. Bacterial growth is not increase in size of the cells. It is increase in the number of cells, number of bacterial cells. That's what bacterial growth is. And I told you that the growth curve occurs in 4 phases: the lag phase, the log phase, the stationary phase, and the death phase, which is the decline. The lag phase, lag is spelled lag. Lag phase is when the cells are very active, but there's no cell division. That's when they are trying to adapt. That's when bacteria are trying to adapt in that particular area, trying to get used, trying to study the environment, try to understand the ecosystem. Okay, so that's the adaptation phase. Then the log phase is also called the exponential phase. So do you know what happens here? What happens here is that there is a very rapid cell division. The bacteria starts to divide at a very constant rate, like the speed. All the cells starts dividing at a particular speed. Like if one is fast, every other one will be fast. That is the log phase, exponential phase. It's more like a very rapid growth phase. Okay, and it's the most active phase. It's even more active than lag. Lag is more like may we just dey observe, may we know wetin dey sup. All right, nothing is going on, no cell division yet. But log phase, there is cell division and it's very rapid. Okay, and then well, then after that, of course, that's where we have the stationary phase. Okay, that's where you have the stationary phase. All the nutrients now, it's as if they are finished exploring, they are finished enjoying themselves. All the nutrients begin to deplete, like they begin to reduce, diminish. All right, they start diminishing in nutrients. At that point now, you now start seeing toxins will now start accumulating. Toxins will now start accumulating. Okay, and this part now is very important for antibiotics. That's when the antibiotics comes, and now the antibiotics now will now start attacking the bacterial cells, and the bacterial cells would actually start experiencing toxins, and the toxins will accumulate in bacterial cells. Then in the last phase, that is the death phase, the cells will die rapidly and they will decline in population. Okay, that is, there will no longer be bacterial growth. Decline in population means that the bacterial cells will reduce. Don't forget that bacterial growth has to do with increase in cells. So increase in cells, so hold your ear, increase in cells is not increase in size of cells. Okay, so whenever the decline phase, that is the death phase, wants to happen, the cell size will not drop. It will just, if they were like 20 bacterial cells, it will drop to 5. Before you know, before you know, no single bacterial cell again, all of them dead. All of them dead. You understand what I'm saying? So that's the point. That's the point. All right. So there are factors that affect this bacterial growth. It's not as if they will just start growing like that. There are factors that affect. Number one is temperature. Okay. Temperature affects. pH also affects. Remember, I told you that there are some bacteria that likes high pH a lot. They like alkaline pH. All right, we also have oxygen. Oxygen is a very huge factor as well. Oxygen causes oxidation, and bacteria likes oxidation a lot. Okay, then we also have nutrients and of course moisture. When they are in moisture rich environment, bacterial cells tends to grow rapidly. Okay, when that particular ecosystem is rich with moisture, it may not necessarily be water, it may be any other form of moisture. Okay, they have that tendency to actually grow rapidly in the presence of moisture. Okay, so that's one thing you must take cognizance of. So so that's one thing you must take cognizance of. All right, then there are some questions that are commonly asked when it comes to bacteria. Okay, bacteria related questions. So I'm going to be telling you those questions. The initial stage of bacteriophage infection. What is bacteriophage infection? Is generally the of course the infection by bacterial cells. All right, what's the initial stage of bacteriophage infection? It's called the eclipse phase. All right, it's called the eclipse phase. That initial phase when someone becomes attacked, all right, becomes invaded by bacteria, is called the eclipse phase. It's called the eclipse phase. It just and at that phase the growth curve will now be log, will now be exponential. All right, will now be exponential, where the cells will start dividing, and of course it will start expanding and all of that. Now the cells will start dividing, and when I say expanding, I'm not talking about in size, I'm talking about expanding in area. They will now cover a large area. That is, there will be many cells in a large area. All right, so that's it basically. Now another question. Okay, the time taken for bacterial multiplication and release is called dash. Okay, that is bacteria to actually increase in their number. Okay, what is the time taken? The time taken is called the burst time. burst, burst time. Burst time. Okay, burst time. So another question is, the shortest time required to kill microbes is dash. What's the shortest time required to kill microbes? What's it called? So it's called thermal death time. Thermal death time. Thermal death time. That's the shortest time required to kill microbes. Okay, so that is that. Now you see these microorganisms and all of that. If we want to actually prevent them from our immediate environment, okay, we need to practice what we call sterilization. Okay, in medical facilities they have medical equipment. So you sterilize these medical equipment and prevent them from experiencing bacterial invasion. So we are going to talk about sterilization because there are lots of questions to under sterilization, okay, that you must actually familiarize yourself with. So I'm going to explain the concept of sterilization. We are just going to discuss it, okay, together. Now, when it comes to your exams, okay, there is a very common definition that every microbiologist knows sterilization to be. Very simple definition. And sterilization is actually the complete destruction or removal of all forms of microbial life, okay, including spores and viruses. So the process of completely destroying and removing all forms of microbial life. What is microbial life? Microbes, which includes spores. Spores is spelled spores, which includes spores and viruses. So the complete destruction or removal of all forms, that is any microbe at all, regardless of the size of the microbe, regardless of the nature of the microbe. Okay, so when you destroy and remove all forms of microbe, what you are doing is sterilization. Okay, what you're doing is sterilization. Now see, I want you to understand, sterilization is different from disinfection. Sterilization is different from disinfection, so I'm going to be defining disinfection as well. In fact, there are some common terms that you need to understand. See sterilization, still see disinfection, antiseptics, all right, germicides, then which other ones? Sanitization. Yes, you see these five terms. Let me repeat them: sterilization, disinfection, antiseptics, germicides, and sanitization. Okay, they have a lot in common, but they are entirely different. So I'm going to be differentiating them. I've already told you about sterilization, and I said that sterilization is when you completely destroy or remove all forms of microbial life. Okay, that is sterilization. Then what is disinfection? Disinfection is the process of killing or removing pathogenic microorganisms. Okay, pathogenic what? Micro, but not necessarily spores. All right, spores are actually very very dangerous when it comes to um inflicting illnesses and diseases. All right, so when disinfection is just killing and removing pathogenic microorganisms. Well, what are pathogenic microorganisms? Pathogenic microorganisms are those microorganisms um that can cause diseases. All right, it's not all microorganisms that can cause diseases, so I want you to note that it's not all low. Sterilization is destroying all microorganisms, whether the ones that can cause diseases, so or the ones that cannot cause diseases. They don't care. When you sterilize, you are killing anything microbial life. But when you're disinfecting, you're only killing and removing pathogenic microorganisms, because there are some in fact all disinfectants only react to pathogens. Disinfectants react to pathogens. A disinfectant will not kill a microorganism that does not cause diseases. I want you to note that. All right, it will not kill a microorganism that will not cause disease. It will only kill microorganisms that can cause diseases, and those microorganisms that can cause diseases are called pathogenic microorganisms. Don't forget that. All right, then we also have what we call antiseptics. Antiseptics are chemicals that are applied to living tissues to inhibit or kill microbes. These ones, they are added directly. Antiseptics, they are added directly to tissues, living tissues. Okay, so you'll be able to kill. This one is not surface something, no, it's directly into tissues. Okay, to kill what? To kill microbes. All right, to kill microbes that are in those tissues. Okay, to kill microbes that are in those tissues. And we are not talking about just pathogenic microbes. We are talking about all microbial life, but this one is the one that is present in a tissue. The one that is present in a tissue. Okay, now we also have what we call germicides. So what are germicides? Germicides are substances that kill microorganisms. This one generally, broad term, kill microorganisms. Generally they are germicides. Okay, like it's germicides that are actually used during sterilization. Okay, they are the chemicals that are used during sterilization. Germicides are the chemicals that are used during sterilization. Okay, although there are machines that are used for sterilization, but when it comes to the chemicals that are used during sterilization, they are called germicides. So germicides, they are responsible for killing all microorganisms generally. This one is like a broad term. This is like broad term. All right, then we also have sanitization. Okay, sanitization is actually the process of we know now, the process of cleanliness and hygienic habits that reduces microbial load to a safe level. Sanitization is not like sterilization. No. Sanitization does not assure you of 100% eradication of microbes. So it is you eradicating microbes to a safe level. Okay, to a level where it is no longer disastrous, or is no longer dangerous, or is no longer life threatening. A safe level. All right, a safe level. That is sanitization. So, but then we have methods of sterilization. Okay, we have methods of sterilization. The first method that you need to take note of when we talk about sterilization is heat sterilization. Heat sterilization. So heat sterilization is the, remember I told you about the fact that sterilization can actually be done with machines, and the most common machine that is used for sterilization is called the autoclave. It's called the autoclave. That is the most common machine used for sterilization. So heat sterilization actually uses steam, like moist heat. Moist heat. So when you say moist heat, you are talking about heat that has moisture in it. Moist heat under pressure. Okay, moist heat under pressure. Okay, like hot, very hot water. Very hot water, or hot steam. Okay, so um basically when you use like what I just said, hot steam, which is water vapor, and you are applying it on a particular um, on a particular material or a particular object, especially all these objects that are used in surgical procedures like scalpel, okay, like scalpel and all of that. You see, those equipments are properly sterilized before they are used, because you cannot just use an infected or um, a bacterial infected equipment to just go ahead with a surgical procedure. You're talking about inside someone's body. You're talking about a particular area in the body system or the tissue and all of that, or organ, and you have not sterilized. No, now it's a medical failure. It's a medical failure. So you must actually use you must actually use that, which is what we call, like I said, a sterilization machine, which is the autoclave. Can you hear me? Can you hear? Someone is saying I can't hear you again. Can you hear me? If you can hear me, say you can hear me. Okay. Then why is okay? You can hear. Why is someone saying the person can't hear me? Who else can hear me? If you can hear me, say you can. Then what's the problem? What's the problem? Okay. Okay. All right. So it's probably the person's network. Probably the person's network. Let me continue. So where were we? Say we are talking about sterilization, right? So I was talking about the different methods of sterilization. So I talked about heat sterilization, where I talked about using steam under pressure, and example is autoclave. Then we also have the temperature is very important. The temperature is very 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 hot. So the temperature that the autoclave uses is 121 degrees Celsius for 15 minutes. For 15 minutes, 121 degrees Celsius for 15 minutes. Okay. So what happens is that when you sterilize, it kills all forms. It kills bacteria, it kills spores, it kills viruses. Okay, it kills spores, it kills viruses, it kills bacteria. All right, it kills all forms of microbial life. I told you, when you sterilize, it kills all forms of microbial life. Okay, then we also have still under heat sterilization, we have dry heat. Okay, this one uses hot air. This one is not moist too. This one is not moist heat, hot air. So example is oven. So an hot air oven, like oven uses hot air to sterilize. Okay, hot air to sterilize, that's an example. So it's commonly used for like glasswares like pipettes, burettes. Okay, used to sterilize pipettes and burettes and all of that, and some metal instruments as well. So some metal instruments as well. Okay, so that one is dry heat. That one uses dry heat. It's still a component of heat sterilization. Okay, then there is also another type of or method of sterilization. It's called radiation. Okay, radiation. So this particular one is a very serious one. So it's like an ionizing radiation, more like it produces radioactive waves, like radiation waves. So this one is that the kind of energy in it is radiation energy. Okay, so what happens is that there's a very high energy here, and it destroys DNA. This one can destroy the gene itself. Just imagine that the microorganisms DNA in the nucleoid is being destroyed. It means that microorganism cannot even regenerate again, because the genetic material has been destroyed. So this ionizing radiation is used to destroy DNA. Okay, used to destroy DNA. Then the non-ionizing radiation is ultraviolet. Okay, that one is ultraviolet. It's also called ultraviolet radiation. So this one does not destroy, it only damages it. It only damages the DNA. Okay, it only damages the DNA, and it does like surface sterilization. It does not go inside. It doesn't have as much energy as ionizing radiation. This one does not have as much energy as ionizing radiation. Okay, this one damages the DNA. It does not go deep to destroy the DNA, including all the double helix and the DNA components and all of that. Okay, so it doesn't do that. So radiation, don't forget radiation. We have ionizing radiation and we have non-ionizing radiation, which is also called ultraviolet. And I said that ionizing radiation has a very high energy that facilitates the destruction of DNA. Okay, while non-ionizing radiation damages the DNA and causes only the surface to be sterilized. It doesn't go deep. Okay, then we also have filtration. Filtration is the third method of sterilization. All right, filtration. What is filtration? Filtration is actually used for liquids that are heat sensitive. This one is talking it talks about liquids. It sterilizes liquids and removes microorganisms from liquids. All right, so this one removes microorganisms from this by filtration. Okay, by filtration. So what do you do in this process? You use some chemicals to filter microorganisms by removing microorganisms by filtration. Okay, you use some chemicals and you use some filtering agents to filter out microbes. All right, so these microbes you would filter them out, not necessarily that you would see them, but they will be filtered out. Remember, microorganisms most times can only be seen or viewed through the microscope. All right, so this is still a chemical process. So it's still a chemical process of sterilization. All right, so it's important, very important to take note of that. Okay, very important to take note of that. So there are also some factors. So there are also some factors that can affect sterilization. It's not as if sterilization is so smooth. There are factors like temperature. All right, like I told you, remember we talked about, for instance, moist heat. The temperature has to be high. Imagine 121 degrees Celsius. It's not play. uh Temperature, even the time too is also a factor. The duration of your sterilization is very important. Remember, we say 121 degrees Celsius in how many minutes? 15 minutes. So time is actually a factor too that affects sterilization. Okay, moisture, the microbial load too. Microbial load talks about, remember we talked about sanitization and all of that. Microbial load has to do with the amount of microbes. All right, so if the microbes are too much, if the microbes are too excessive, there's a tendency that it would require more time to actually eradicate, or to actually be eradicated. Okay, to require more time. Okay, so now there are some questions that are commonly asked. If you check your past question, you see this question: which is a method of moist heat sterilization? Which is a method of moist heat sterilization? The answer is autoclave. Okay, now hot air oven is used for sterilizing dash. Hot air oven is used for sterilizing dash. The answer is glassware and dry materials. Glassware and dry materials. Now we also have what we call endospores. Endospores are destroyed by dash. Endospores are also microbes, are also microbial life. So how are they destroyed? They are destroyed by using germicides or sterilization. Germicides or sterilization. All right, then drugs effective against mycobacterium. There's what we call mycobacterium. It's actually a form of bacteria that is in some common bacterial diseases. It's a kind of bacteria that causes some common bacterial diseases. So sometimes this mycobacterium is usually in air. So what happens is that it enters the body system through inhalation when you're breathing in bad air that contains mycobacterium. Now it's actually important to use germicides to eradicate them before they even um enter the system, the body system. But when they are inside the body system, you use drug effective. Okay, you use drug that are effective against this mycobacterium, and they are also called germicides too. So we have external germicides and we have internal germicides. The internal germicides are the drugs. Then the germicides that you fleet in the environment to kill germs, they are called the external germicides. All right, the drugs that you take in, the antibiotics that you take in are germicides as well. All right, are germicides as well. So that's very important to take note of. All right, so in tomorrow's class we'll talk about viruses. Okay, we'll talk about viruses, we'll talk about it in details. All right, to talk about viruses in details, I'll also ask you, we'll also look at some exam questions on viruses. I've been able to explain fungi, all right, in details, but I'll also be explaining some commonly asked questions under fungi, frequently asked questions under fungi tomorrow as well. Then we'll also talk about some things in staining. We'll talk about some things in staining. We'll talk about culturing. Then we'll talk about which other thing did I say? We'll talk about those culturing techniques. Yes, culturing techniques. Then we'll talk about culture media. All right. So we'll talk about all these things in tomorrow's class. So don't miss tomorrow's class, because after every class we'll try as much as possible to answer exam question, just like we did as well today and as well as yesterday. All right, so don't miss tomorrow's live class. Don't miss tomorrow's live class. So this is where we would call it a day. This is where we call it a day. This is where we end today's class. All right, so do well to, you can actually refer back to this particular um, session anytime. Okay, it's there on the link is available there on YouTube. You can make reference to it and do your revision as well. All right, so that is that. So this is where we would end tonight. So see you in the next live class. See you in the next live. I believe you can hear me, right? I believe everyone can hear this farewell information as we end. I believe you can hear, right? So just if you'll be available for the live class. Okay, just okay, good. I can see your responses. Okay, I can see your responses. So that is that. Always revise your notes and always go through them. All right, go through them. You cannot be a good student without reading, without studying effectively. All right, don't give any room for laziness, don't give any room for negligence. Okay, and so I will see you at the top. I will see you at the best point. I will see you at the victorious point. Okay, so have a good night rest and see you in the next live class. Bye.